Hey, it's 7:05, so let's get started. First of all, welcome everyone. Thank you so much for coming to Darwin Day 2021, hosted by the Halton Peel Humanist Community and Cafe Scientifique Mississauga. Darwin Day was actually Friday, February 12th, so we're a little bit late, but we're still celebrating around the right time, and that marks the 212th anniversary of Charles Darwin's birth. Obviously, he is the famed originator of the theory of evolution by means of natural selection. And I find Darwin a very fascinating person because, first of all, to see his writing, I had the chance to read On Origin of Species last month. And, he really captured a lot of the core of the theory of evolution as we know it now and spoke about it in such a sure way that he had managed to assemble all this evidence for his theory, and it's amazing, and it now kind of makes sense. I thought, with it being kind of a very basic idea, how was he being compared to other key figures of science such as Newton and Curie and Einstein? And that in itself is curious. Why do we have a couple famous scientists that we choose to celebrate? And I think that's because it's important to have personalities, to have people that we can connect to, so that there's that human connection, and we can really understand that science isn't just an abstract concept. It's something that we in our everyday modern lives can potentially do. And by having those personalities, those people who did science and continue to do science, we can inspire ourselves to be part of the next big discovery. So speaking of people who do science, I'm pleased to announce our speaker for tonight, Dr. Catherine Beauchemin. So she is a professor in the Department of Physics at Ryerson University in Toronto and a deputy program director for the Interdisciplinary Theoretical and Mathematical Science at Riken in Walco, Japan. Her research interest is in self-organizing systems, particularly in mathematical and computer modeling of infectious disease dynamics. Obviously, a big topic of discussion for the past year or so. Today, Dr. Beauchemin will be sharing with us how, through mutations and genetic reassortment, a virus can mutate, and the resulting virus variants can evade our drugs, our vaccines, and our body's own immune response. As I alluded to just a moment ago, this also is a topic of much discussion these days, with the different variants of SARS-CoV-2 being front and center in the news. So with that, I will hand it over to you, Catherine.
So thanks for having me. Thank you everybody for choosing to be indoors tonight. Although if you're in Canada, if you're in Toronto, there was not a tough call. But thank you for spending it with me. I know your entertainment options are many with Netflix at the ready. So I want to start with my disclosures, and my financial disclosure is that in the past I have received consultant fees and research funds from the pharmaceutical companies F. Hoffmann-La Roche, AstraZeneca, and Adamas Pharmaceuticals. But currently my salary and all my research funding are from public government sources only. I also want to make an expertise disclosure with that I'm a physicist and not a virologist or a microbiologist. And in fact, I have no biology training past high school in theory, but in practice, my research is in the construction of computer and math models to capture virus infections. And in fact, I've been working nearly exclusively on influenza for the last 17 years. So I can hold a candle to any virologist on influenza and a number of other viruses by now. So I wanted to start this talk by maybe establishing a lingo so that when I use words, we all know what they mean, because we hear them often and people are far more comfortable with technical terms in biology than in physics. People immediately are worried about physics terms. And I find that people more easily use biology terms like amino acid. They'll tell you, oh, this shampoo is full of amino acid, and they use it to make you think that that's a good shampoo, right? So the point is, I thought it would be useful to, instead of just using these words as complicated things that mean nothing, I would start by maybe defining what they mean. So basically, we'll start with proteins. So proteins are basically the structural building block of viruses. So it's like the Lego blocks of viruses. And for example, if you think of the hemagglutinin on the surface of a flu virus, or the spike protein on the surface of the SARS-CoV-2, which is the virus that causes COVID-19. So if you look down at the bottom, I have a picture of an influenza virus. It's obviously enlarged. And so you can see this spike is called the hemagglutinin. And so that would be one protein. And what a protein is, it's a very long chain of amino acid, and it folds itself into that shape. And given that shape, it can perform certain function. Now, there's also the word RNA. We've heard a lot, especially in the context of mRNA vaccines. No, I don't need the quotes. They are mRNA vaccines. So the RNA stands for ribonucleic acid, and it's a string of monomer units. So it's a polymer chain. So it's basically large molecules, and there's a string of these large molecules. So molecules are made of a bunch of atoms. So you have a bunch of atoms assembled into a molecule, and you have a chain of molecules, and that would be your polymer. And it's made of 4 types of monomer. There's the guanine, the uracil, the adenine, and the cytosine. So the GUAC, you've probably heard of those bases for DNA. It's GTAC, and they've got pairing. So this is the same idea, but you have a U instead of a T. And so from those, you have these long string of GU of this alphabet. And if you have 3 of them, these 3 can form a codon. So a codon is 3 ribonucleotides. So this is 1 ribonucleotide. So 3 ribonucleotides, which get translated into one of 20 amino acids normally. So for example, if you have U, A, and U, that would be translated to an amino acid called the histidine, or HIS for short, or just H in the 1-letter format. So both UAU and UAC make a histidine. But if you replace the U with an A, you get a tyrosine. And so that's the way in which you could get one mutation, and you go from having one amino acid called histidine to another amino acid called tyrosine with this one mutation in that one nucleotide. But some mutation won't do anything, right? So if you replace the U for a C, you're still having a histidine, right? So that would be kind of a silent mutation. It doesn't manifest. There's also some codon that don't code for an amino acid. For example, these 3 sets of codons actually is the stop codon. It means stop reading. So if you're reading along, stop reading when you get to that code. And so this is kind of a way to picture how that works. So if you had this messenger RNA, which we've heard so much about, so it will have these base pairs. And what's going to happen is that some tRNA will get assembled that matches this basically triplet of letter, this codon that matches, that complements the codon on the messenger RNA, and it will have loaded on it the amino acid, the next amino acid in the chain that the messenger RNA is spelling out. And so over time, you have more and more of these coming in, and you can form this long, long chains, this peptide chain. And if it's long enough, then it's a protein chain. And so you have these very long structures of beads, and eventually those beads, because of their property, will fold and take on a specific shape. And it's those shape that can then perform a function on top of the virus. So we've got our base language established, and that alone took a large fraction of the time, but I think it's important that we start putting these concepts together. I also wanted to talk about all these words that have been thrown about, but specifically in the context of viruses. So the word mutation, so now we have a bit of a sense of that. So if you had this triplet codon, CAU, and you replaced the first C with a U, you went from a histidine to a tyrosine. So that's what we call a mutation. So that's one mutation, a single site mutation. And that would make a variant. So you now have not the original virus, you have a variant of that virus. A variant is a virus that has not enough mutation or not a significant enough mutation to be called a strain. So a strain normally would be a set of mutation that characterize a group of variants that altogether are sufficiently different to be considered a separate strain. And all that is completely arbitrary-ish. It's usually decided by committees. There's no hard and fast rule when it comes to viruses for defining what is a strain. And for example, the influenza community might take very different decision than the SARS-CoV-2 community. So it's very decision by committee here. There's no formal definition of a strain. Normally, though, what you're aiming for when you name something a strain is really a property. This set of mutation tend to give this specific property to the virus, more transmissible, more deadly, and then you would want to bundle all these sets of variants that have those mutation as a strain. And then there's lineage, and lineage actually means ancestral genetic variant, right? So it means you have a set of variants or strains that are, and here's the key word, believed to have come from the same ancestral genetic variant. So if you're finding them in nature, these various variants, and you want to decide whether they're all from the common ancestor, you could do it kind of by inference. So it's got that many distance, so it's more likely to be related to this than to that, and so on and so forth. But it's not always possible to do that. For example, a mutation, a set of mutation could emerge completely separately by two separate events by chance. And so it's not because one is just one mutation away that he's necessarily related. So oftentimes you'd need additional information like, where did this happen? Where did you get the virus? And that would be helpful in establishing lineage. So because of all that complexity in a human, establishing lineage is much, much simpler. But for viruses, a lot of these concepts get very confusing, and that's why they're not absolute and they're very blurry. And in particular, the CDC reminds that the press often use the terms variant, strain, lineage, and mutant interchangeably for the time being for the context of these variants. And they're talking about the COVID variants here. The first three of these terms are generally being used interchangeably, even by the scientific community. So that's what you should know about that. We can't get that nitpicky when it gets to viruses, especially developing viruses. Okay, the next thing we need to cover in terms of our basis is what is a virus infection? And it sounds obvious, but it's worth walking through the steps. So again, I have my flu virus here, and you can think of it as having two kind of main parts. There's the structure, so what it's built of, and then there's the instructions in the middle. So these little squiggly green things are the RNA segment, and there's a total of 9 separate RNA segments packed into a flu virion, and each of them encode for different stuff. So when the virus enters the cell, so this big square rectangle is the cell. So this is virus replication viewed by a physicist. So you can imagine there's kind of 2 parts to this. There's the structure of the virus, and then there's the instructions. And so the first thing that's going to happen is these instructions are going to have to be transcribed. Some of them will be transcribed into messenger RNA, and some of them will be transcribed into complementary RNA. So the messenger RNA, and again, some viruses are DNA based, so I'm talking here specifically about flu, and different viruses will have small modification on what I described. So here the messenger RNA will go on to be translated into protein, so make more of that virus structure. And the complementary RNA will go on to be replicated to make more of that initial instruction that came in to be packaged in a new virus. And then through some magical, very complex dance, the structure and the internal instructions have to meet up at the end in the right proportion, in the right place, and then get packaged into new virions that go on to infect other cells. Now, why are viruses a problem? Why do we care? So we care for a number of reasons. So if the virus, as part of that replication, so the reason why the virus can replicate to begin with is because the cell is perfectly happy when given a set of genetic instruction to make the recipe, right? It just like it gets it, it makes it. That's what happens. And if it's busy making virus because the virus has utilized that pathway, it means that there's kind of less recipes to go around for other stuff that needs to be made by the cell for the cell to do its usual job. And so if it's a hard cell, that could become a problem fairly fast. And so this is one way the virus can interfere and eventually cause death, or at least cause significant unpleasantness. Another thing is the virus itself, through the replication, can produce toxic products that would be toxic to the cell, and then the cell will actually die from that. Another thing that can happen is so many viruses get made that they burst the cell. And so that's another way you can lose your cell. And then the other thing that can happen is your immune system itself could actually be the result of your death. So without your immune response, the virus goes on to infect other cells, to infect other cells, to infect other cells. As all these cells lose function, you'll die. And so it's important that your immune response kicks in. But sometimes if it kicks in at the wrong time, too strongly or incorrectly, it could actually be what kills you. So it could come in, flood the area with tons of cells and go in and trash the place. And it's great, they've stopped the infection, but they've actually destroyed a whole bunch of tissue, and now you're drowning in your own lungs, right? So that could happen. And so this is other ways that you can die from a virus infection. So I think I've covered most of the ways a virus infection can kill you. So I'll go on. So let's talk about our first virus that I want to talk about, and is the one I'm most familiar with, and it's influenza. So it's among the 10 leading causes of death in Canada. And each year, in a normal year, it'll cause about 12,000 hospital stay and 3,500 deaths. Comparatively, COVID-19 since February 2020 has caused approximately 21,000 deaths as of February 14. So when people say it's just as deadly as the flu, it's just not true. Also, I'll have you note that we had to all lock up in our rooms to keep it at that low number of 21,000. And so you can imagine if we were doing nothing about it, how much worse it could be. And therefore the statement, it's no worse than flu, is just absurd. The other thing that you should know about influenza is that it's often separated into different genera. And in particular, you'll have heard of influenza A and influenza B most importantly. So influenza A is most important because it's usually the one where pandemic comes in. So there's 1918 pandemic. There's also the 2009 pandemic, the H1N1 pandemic that I'm sure everybody remembers. And in influenza B is more rare. No, it's not more rare. It's less likely to cause severe illness and death, but it can get bad when it gets into a residential community like a nursing home. So the way you identify a flu strain is as follows. So first you have the flu strain, so it's influenza A, the place it was isolated, in this case, California, the lab sample number, the year, and then the subtype, if it's an influenza A. So H3N2, H1N1, H5N1, H7N9. So these are the subtypes of influenza strains. For influenza B, we don't use subtypes, we use lineage. So there'd be B Phuket, isolated in Phuket, but here instead of the H1N1, we say Yamagata lineage. So it would either be Yamagata or Victoria. So those are the two lineages that influenza B comes in. So that's how we identify these strains. Now, we get to focus on a less physicsy looking picture of virus replication. So this is specifically for flu. So you've got the flu virus with its 8 segments. It comes in, it fuses with the cell, the virus inside gets fused with the cell inside, and these little segments get off-packed. And here you are with your viral RNA. And some of it will be transcribed to mRNA, which will go on to make protein. Some of it will be transcribed to cRNA, which will go on to make more of these little segments. What's important is that all 8 of these segments need to independently do this and be successful for you to have another virus at the end. So that's a kind of a tricky trick that you need to get all of those to make it, to not get degraded, not be eaten up by the cell, and for all but 8 of them to get packaged at the other end in exactly the correct ratio. So it's not a simple process, and I'm not going to go into those details, but those are some of the difficulties that the virus has to face. The other thing I want you to notice is that you can see the virus is assembling at the cell surface. And so in fact, the envelope around the virus is actually your own cells. So it's used up your own cell surface to make its coat. And I'm saying that now because it's different for SARS-CoV-2. So I also want to talk to you a bit about the antiviral drugs against influenza that exist. So some of them, so the adamantane, so amantadine and rimantadine, they block that fusion event where the virus can unpack its viral genetic material. There's also polymerase inhibitor, and what they do is they prevent that transcription stage from happening. There's also the neuraminidase inhibitor. So when influenza comes in, so when the virus comes in, it first needs to attach to the cell surface. And for the case of influenza, that's the hemagglutinin. The H of H8, like H1N1, the H is the thing that attaches to the cell surface to let the virus in. Well, on the way out, as the virus is trying to leave, the H is still attaching. And so if the virus wants to leave, it needs to do something new. And so what happens, the neuraminidase, the N of the virus, so remember H1N1, that's the N of H1N1. So the N is responsible, it's like a pair of scissors. It will cut the link between the cell and the hemagglutinin and allow the virus to break free. So a drug like Tamiflu, what it does is it sticks itself in the scissors, the neuraminidase scissors, and prevents that cutting from happening. And so the virions are still produced, but they can't be released from the cell and go on to infect other cells. And those mutations that can happen when the virus replicates can cause drug resistance. So for example, there's a mutation called the S31N mutation that actually cause adamantane resistance. So this is the fusion inhibitor. So that mutation in the M2 protein, because that's what it blocks, actually prevents the virus from acidifying. But that drug had to be abandoned in 2005 because the S31N mutation was at 100% in nearly all virus backgrounds. So that drug became largely ineffective for influenza. And for example, in 2018-2019 flu season, it was still at 100% in all H1N1 and H3N2 strains. It means 100% of the strains were resistant. They were not 100% resistant. So I just want to clarify. And in the Tamiflu, that resistance varies from year to year. It was 90% back when we had the 2009 pandemic of H1N1. And it's now down to about, it was 0.4% in the 2018-2019 flu year. Something I should mention, I want to make sure I don't forget, because this year everybody is staying indoors, and because SARS-CoV-2 is so much more transmissible than flu, this year the flu is basically a non-issue. It's nearly non-existent. And I think it could be very interesting what the consequence of that will be for the strain landscape. Because you have all these strains circulating all those people when you have a big flu year. And now you're down to nearly nobody infected with the flu. So is it still around? Might we wipe out the flu? Possibly not. It is still around, but it will be interesting to see whether the strain landscape completely changes on the other side of this pandemic, if we can all hope that there's another side. So you might ask yourself, maybe that's come up, why do we need a flu vaccine every year? So it's not actually because we lose immunity over time. It's because the virus is changing. So every year you're still immune to the strain that you got the vaccine for, but the strain that's now circulating is a different strain. And that happens through two main processes, one called the antigenic shift and another one called antigenic drift. And the antigenic shift is due to mutation and antigenic drift due to reassortment. So I'll show you what I mean. So the antigenic drift, so that's the mutation. So you can imagine one of these segments in the flu virus still, it's got this long segment and it needs to be copied. So part of what happened is transcription. So this is the virus's own polymerase. So it gets packed with that. These little three balls run along the little yellow balls and they make a copy of it. And it's a positive sense copy. And you can see that segment gets longer and longer as the little balls get further along the chain. And it could make mistakes as it do that. So that's one way that mutation can be introduced. Similarly, when you're making the CRNA, from the CRNA, you're making the new templates later. These little balls run along the right string and can make these new segments that are going to be packed in a new virions, and there could be a mistake there as well. So this is mutation. So these would accumulate over time. So that's why it's a drift as opposed to a shift. So we'll see now what a shift is. So an antigenic shift, so don't forget the virus has 8 segments, and so for flu. And so you have, let's say this virus here and this virus here, and both of them infect this duck, and in particular, infect the same cell inside the duck. What could happen is because all the segments independently replicate, we could end up with 3 segments from this virus over here and 5 segments from this virus over here. And so we have some viruses coming out with various assortment of orange virus and purple virus stacked into the new viruses that come out the other end. And so that would be a huge change. And so that can happen in the bird, it can happen in the human, but here's the problem. The pig can actually be infected both by bird viruses and by human viruses. So what could happen is a human virus and a bird virus infect simultaneously a pig. 2 of those viruses end up in the same cell, and we end up with a bunch of segments from a bird virus and a bunch of segments from a human virus. And then we can end up with this mutant virus that could have all the lethality of an avian strain, but all the transmissibility of a human strain. And that could be rather dramatic. So these are the kind of event that could cause the next pandemic. And to be frank, I think this is a big concern for everyone working in this field, and something people are keeping a very close eye on. And so those events are called reassortment. And so because of antigenic drift and antigenic shift, we need to continuously reformulate the vaccines. And you might wonder, how do they do that? Well, so influenza is highly correlated with winter, and there's a northern hemisphere winter and a southern hemisphere winter. So what scientists do is twice a year, they meet and they decide on what will be the vaccine for the following flu season in the winter. What they do is they look for us, for our northern hemisphere vaccine, they look to the southern hemisphere and see what are the dominant strain in the southern hemisphere that winter, for their winter, and they use that to decide what should be in the vaccine the following year. So what ends up in your vaccine? So this is tough. You have to make a guess, and you can only pack so many of those strains. So let's look at how the flu vaccines are packed. So there are 3 types of influenza vaccines you can get in Canada. You can get the trivalent or the quadrivalent, meaning 3 strains or 4 strains. and these are the intramuscular ones, are the inactivated influenza vaccine, or you can get the quadrivalent live attenuated vaccine. You'll know immediately which one you got because the inactivated one is a shot, and the live attenuated one is a spray that goes up your nose. And so you will definitely know which one you received. So for example, in 2018, 2019, Northern Hemisphere flu vaccine, these were the 4 components. If you have a quadrivalent, if you had the trivalent, you only receive the first 3. And if you got the quadrivalent, you receive 4. And what you see is how it's packed every year. They choose 1 representative for H1N1, 1 representative for H3N2, and 1 representative for flu B. And if they have a quadrivalent, they can pick 2 of flu B. So that's how it's always structured. So 1 H1N1, 1 H3N2, and 1 or both of the flu B lineages. So that particular year, they had picked these 4 representatives. And it turns out that the one they picked only accounted for 56% of the H1N1 that were circulating in the end. Later, like after they decide on the vaccine, in the end, 56% of the strains were similar to this one, which means your immune response would have been responding to the because of the vaccine better to that strain. So there would have been protection, but only in 56% of the case. For H3N2, it was 97% of the circulating that were like the one you got in the vaccine, which is good. For the flu B, if you had the trivalent, you only had 19% protection. If you had the quadrivalent, it adds up to, what is that, 30%. So you had 30% protection for the flu B if you got the quadrivalent. But you can't just look at that independently. You have to also ask yourself, but what proportion of the flu that was circulating, what of each type? So that year, you had 66% were H1N1, 29% were H3N2, and 5% were flu B. So if you multiply the protection you get times the prevalence of the strain, you get the estimated average efficacy, which was around 67%. So not a great year. And mostly it's because of this screw up with this guy's a problem. And also the flu B were not a huge success. But we did very well on the H3N2. It just was not very prevalent. So that's how these vaccines work. And so every year they have to do their best. So then you might ask, given that, is it worth getting the flu vaccine? And in particular, is it worth getting it every year? And there's been 2 studies that have tried to look at this problem. And you will not be surprised by now, having read all the COVID stuff, that the studies don't agree. So the first study said that it's not protective in the long term. And the second study said that it provides continual protection. So the opposite. But actually, if you look closely, you can actually explain this through a phenomenon called antigenic distance. And specifically the antigenic distance between the first vaccine you received, the vaccine you received, for example, in the following year, and what strain is circulating that year. So when I'm talking about antigenic distance, what I mean is if you have that virus and you have these 3 mutations, then you are at an antigenic distance of 3. It's a bit of a simplification, but it's a good way to think about it and to conceptualize it in your head. So let's see. So we have the vaccine. And what it does is simulate antibodies that are similar to it. And the further away you get from an antigenic distance space, the less response you get. So you get a big density near the vaccine strain. If you get your second vaccine the second year, so this is an example of distance that could exist, what's going to happen is instead of getting a big, strong response here, you're instead going to re-stimulate the ones that you had in common. So instead of concentrating your strain, your effort, your immune system here, you'll be concentrating it further over here. So enters the actual infection. So let's say you're exposed to what's circulating. If what's circulating is here, well, that's great because you're going to get protection from these original antibodies you got from vaccine 1 that got re-stimulated by vaccine 2. But if your circulating strain was over here, you're in trouble. Because even though you would have been well protected by vaccine 2 only served to stimulate this space. And what that means actually is that you're left unprotected by the vaccine as a result. So having had vaccine 1 caused vaccine 2 to be ineffective, which therefore caused your exposure to be equivalent to you having never received the vaccine. There could be worse case. So this is a not best case scenario because you're not protected, but it's not worse as in you're not left any worse than if you had not received the vaccine. So on average, you'll do better than not having been vaccinated. But this is an example of if you look over a bunch of years where this phenomenon is repeating over and over again, then your conclusion over that set of years is that overall it's not a good idea. And that's specifically when they look back at these two studies, they were done over two different set of years, which is why they came with a different conclusion. That distance was not the same over this set of years. So that's how sometimes science can disagree. And so now you know. So I'm going to start talking about the virus that you're possibly most interested in. And I have maybe about 10 minutes to tell you everything about it. So the first thing I want to tell you is that the virus itself is called SARS-CoV-2. And it's this virus that is causing the disease. So COVID-19 is the name of the disease. SARS-CoV-2 is the causative agent. And that's what he looks like. And we've seen it, plenty of him. It was most likely the third leading cause of death in Canada from February 2020 to March 2021, because it's been responsible, as I said, for 20,000 deaths over that period. And so that would put it behind cancer at 80,000 per year and heart disease at 53,000 per year. But the next cause is less. I think it's 13, and I think it's accident. And so actually it's now the third highest cause of death in Canada. Now the SARS most likely, I mean, the other thing that could have happened is maybe more people died of heart disease or cancer because of public health issues. So I'm really comparing with 2019 numbers. So I can't say for sure that it will be the third, but if nothing else changed, it would be. The SARS-CoV-2 is actually part of a genus called the beta coronavirus genus, and it's got a bunch of lineages. And in particular, those lineages, we could call them A, B, and C, but they're called the mbecovirus, the sarbecovirus, and the merbecovirus. And these contain various viruses that have infected humans before. And what I want to draw your attention to is two things. These ones, the HCOV OC43 and HCOV Hong Kong U1. And so these are actually part of the set of viruses we refer to as the common cold. So there are some SARS viruses that are, well, actually, these are not SARS viruses, they're coronaviruses that are not too bad. But we are here with our SARS-CoV-2, and he's actually quite similar to the SARS-CoV-1. So they're quite similar viruses. And so that's why they're in the same lineage and subgenera. Along with these 5 that I'm naming here, there's another 2 that are part of the alpha coronavirus, and those are also responsible for what we call the common cold. Replication, it's a bit different, and I'm going to point to you specifically the ways in which it's different from flu. For one thing, it's got a single segment. So it doesn't have to have 9 segments that somehow have to make it into the replication process. Only a single segment, so you don't lose parts. Not in that way anyways. And so what first has to happen is it needs to make proteins, and these proteins then actually form, they will self-cleave to form this process called the replication transcription complex. And that's the thing that's going to make the other subgenomic RNA. So what happens is you have this very long segment, and instead of having one segment for one protein-ish, as you do for flu-ish, in this case, you have this very long string. And what you do is you read some part, and then that has to be cleaved, and that will be part of one protein. And you read the next part, and that will be another protein. So what you see here at the bottom is the different reading frames on the same long string that encode for different parts of the virus. And so you have here the spike protein, the envelope protein, the membrane protein, and the nucleoprotein. So all of these 4 things, so these 3 will end up on the surface of the virus and will be visible. These ones end up lining the RNA, or the little balls along the RNA, to prevent it from interacting with itself. So first difference, it's a single segment. Other important difference is that the virus actually buds on the inside. So it still borrows your cell surface as an envelope, but not the cell surface actually. It's a side of your endoplasmic reticulum, and the Golgi in particular is what actually ends up forming the surface of your virus. What that means is that inside your cell, if it's infected, there are fully formed infectious viruses. This is different from flu. For flu, because the virus forms at the surface, if you kill an infected cell, all you have is virus bits. You don't have any formed virus. But for SARS-CoV-2, if you kill the cell, you end up with a whole bunch of infectious virus that can be released, potentially. About those variants, I figured you'd want to hear about that. So the main variants that you've heard about are the UK variant, the South African variant, and the Brazil variant, and they go by various names because, as I said, there's no strict definition of these things, and anybody who work with them are free to kind of make up their own naming. And so GSA actually calls it the B117, which we heard a lot referred to in the news. The next strain people call it the 20I501YV1. And you heard earlier on, they were called VOC for variant of concern with a date. And even earlier than that, they were called a variant of interest, VUI, with the date. So it was a variant of interest until it became a variant of concern, until we finally gave them some more complex names. And so they contain mutations, like these varying mutations. And you'll see in particular, this mutation is common to all three of the variants. So it looks like it's probably going to stabilize and be here to stay, this particular mutation. And the S means that it's in the spike protein. So in particular, the UK variant is said to have an unusually large number of mutation. And people believe that this did not happen over time, but rather in a single person. And they think perhaps a long hauler, somebody who's been producing virus over a long period of time, so more of these mutations could accumulate, or perhaps an immunocompromised patient for the same reason. But again, this is what people believe. It's much harder to actually prove these things. The South African variant is believed to have emerged independently of the UK variant. So even though it shares a number of the same mutation, it's not actually, they believe to have occurred completely independently. And the Brazil variant shares a lot of the mutation, again, with the South African variant. And it was first discovered, which I think is interesting, at Tokyo Haneda Airport, because they test intensively when people fly in, and it was discovered on 4 travelers from Brazil. So it's actually a Brazil variant, but it was first detected in Haneda. And the source of genetic diversity for SARS-CoV-2 are a bit different from flu. So there's the individual mutation, which is similar to flu, that can happen, but we can't have this reassortment where one segment end up with another segment. Instead, what it does, because it's got this long single segment, what can happen is if you have a cell that got infected with 2 viruses, you have 2 of these long strands. So what can happen is the virus can, the machinery can start reading along and then make a skip and jump to the other purple strand and start reading from it. So that's how you'd end up reading partly from one and then skipping and reading partly from the other. And so you'd end up with a new genome that is a little bit of A and a little bit of B. And so those are called recombination as opposed to reassortment events. And this is what it looks like. So I'm wary of time. I'll be 2 more minutes, don't worry. And so basically that's what the virus over time looks like. So this is the graph. So I've taken this from the group called Next Strain, and this is available online for everybody. You don't even have to be an expert or have some login credential. The x-axis here is the number of mutations relative to the consensus strain that is 19A. So this is just one long series of letters. And so this is how far apart these various strains are. So you can see that the 19B was two mutations away from 19A. The 20A was 3 mutations away, and so on and so forth. Our variants of concern here or over here, this is the variant of concern 2. So this is V2, this is V3, and this is the Brazil strain, this is the UK strain, and this is the South African strain. And I should not call them strain, I should call them variant. You see, I did it. So at any rate, so this is where they're at. So this is when the x-axis is mutation. There's another way to look at it, which is over time. So this is the number of these variants over time. And so this is February over here. So you can see that some of these lines don't reach out to February. So some of these lines have stopped, and some of these lines are now taking over and filling in. So that's how you can see a shift over time. This is also in the Next Strain website. Anybody can go and look at that. Another fun thing to look at is the strain replacement. So we can see that 19A originally, that consensus strain I was talking about, made up 60% of the strain, and over time got basically wiped out by a combination of different strains that were then 19B, then 20A, and you can see even those are going away, and these are the variants coming in. This is variant, I think this is 3, 2, and 1 in order. So right now those variants are making up for about 30% of the strains that are being sequenced. So that's a concern. It's not a good picture because only rich country can afford to do that. So we don't have a full, this is not a representation of all the strain, it's a representation of strains where we're able to sequence. So it's good for what it's good, but you can see the shift. So if we want to talk about a visualization, a visual reminder of the virus evolution over time, this beautiful rainbow graph is a very good proof of that. And again, that's all available on the Next Strain website. So you should go check it out if you're curious. last two slides, I promise. So how do we end up having this shift over time from 19A to these 20I, for example, and these variants? It's if you have a larger reproductive number. So if a particular strain can infect more people, then if one strain infects 3, but the other strain infects 1, very quickly, the 3, because it's multiplied by 3 every time, very quickly, it takes over, right? It's got an enormous crushing advantage. And that can happen for different reasons. One of them can be that the set of mutation that the strain got is actually allowing it to escape an antiviral drug. We don't really have that for COVID-2, for COVID-19 or SARS-CoV-2, so that's not really a big concern yet. The other thing is that it could be a mutation that allows it to escape pre-existing immunity. And again, we don't have a whole lot of that, but that's coming online, and that's a big concern, as you know. But then another thing that can actually do it is that it could be, even if it has equal transmission ability, if the actual strain is being born in a population that has a very high transmission rate, let's say a group of people that don't believe in social distancing and are all hanging out together. Well, whatever mutations that these people develop will rapidly take over much more widely than if you take a group of people who are staying distance. So there are properties of the selection pressure that are not inherent to the virus and can have to do with your behavior. So when people say, these variants are inevitable and, this and this is going to happen, our behavior has the potential to completely alter these trajectories. For example, what happened to flu in this season, right? The fact that we're all staying indoors to prevent COVID means that we're completely changing in very fundamental ways what's happening to the strain landscape of flu. And the same thing is true for COVID. We could shift it to exactly what we wanted if we all got on board. So that's my take-home message. Basically, that small mutation and large mutation over time can affect the severity of the virus, but also its efficacy of the immunity, the antiviral drugs, and the vaccine. That we can predict virus evolution, but to do that, we need to understand how the genotype of the virus maps to its phenotype, so how the mutation maps to changes in how the virus reacts. And we don't have a good way to do that right now, other than by trial and error. Like if you get the virus, you isolate it and you test. And that's how you know. You can't tell by just looking at a genetic sequence. And math modeling will be essential to be able to do that in the future. And that's what I hope I will be able to play a role in. And that's it. Thank you very much.
All right.
Thank you.
Catherine, thank you so much. Such an interesting talk. And I mean, flu's been around for a long time, but I knew none of that about the different sections of genetic code that could swap and how that was different from SARS-CoV-2. So thank you. So we're going to open it up to questions. And what I'm going to ask is if people could raise their hand.
My question is whether you'd like me to stop the screen sharing to give people a chance to see better? Yeah. Okay.
Yeah, let's do that.
I'll do that.
Yeah, so
now I can see you better.
Excellent. So people in the chat, they're able to, if you put up any symbol, because I don't see an actual raised hand here, then we'll come to you and ask you to unmute. If anyone has any questions.
The raised hand icon is found on the reactions menu, Cam, so just so everyone knows where the raised hand is.
Or people could use the chat, I guess.
I see, is it Gilles? Yes.
Gilles. Thank you.
Apologies.
So, yeah, I'm a colleague of Catherine, actually. Like we are.
Yes.
I'm co-affiliated with the ITEMS Institute at Riken in Japan. And so, yeah, thank you for hosting this and thank you for the presentation. But so, can you come back to like the mechanism by which the virus reproduces itself, like on slides, like slide 7?
Okay, let me bring that up again. I think it'll be easier if I share my.
I'm puzzled by like how does it? I understand that like a cell will like use the genetic material and produce the parts, but like how does it reassemble itself? Like this part looks like magic to me. Like
it is.
Like,
it
do
is
the parts
magic.
know how to like they go together? Is there something conducting it? Like.
I actually don't know. I know some of it, not all of it. So basically, this one is perhaps an easier one. So there are pathways that naturally traffic. So because the cell is used to making proteins and from genetic material, there are normal pathways in the cells that exist. So when this mRNA gets found, it gets taken to a specific place where these things take place for all mRNAs. And so that protein production always happens at the same place. And in particular, it ends up budding those proteins, if they're going to be surface protein, right on the edge of the endoplasmic reticulum. So that there are pathways that control these with a series of like acidification and localization. So sometimes there'll be additional bits that get attached that kind of tell you when the tell mechanism inside the cell, when you see that signature, bring that stuff over here kind of thing. And by bring that stuff over here, it can mean by ATP or by like, there's various ways that things can be trafficked through a cell. And I don't know them very well because I'm not a cell biologist. And so my knowledge is very weak as a physicist, but those are some of the processes that can make these things happen. And then once they're embedded in the endoplasmic reticulum, again, there are processes that actually on a regular basis bubble these out to the Golgi, and then the Golgi actually bubbles things out to the surface. It's just what it does. It's a normal ways by which these things happen. Now, how
and
then?
The genetic code gets bundled with it.
Right, so that's more complicated, and it's not fully resolved. So I don't know a lot about it, but I can tell you that it's not fully understood. And in particular, until recently, people thought that it always packed 8, and it seemed to not be entirely true. So some of it can be attributed to geometry. So these segments are not all the same length. They get attached through this. So you can see all these proteins. So these proteins, when they get made, they're trafficked back. And once they get attached to the segment, it gives the signal to other things in your system that this has to be trafficked to an area. It gets attracted to the same area that these things get released on. So you can imagine that the whole cell, it looks very localized, but in reality, you can imagine that the whole cell surface is just dotted with these things. And so it could just kind of go kind of anywhere. And so these things would pack together and then would end up coming here. And if you can imagine trying to pack one too many long segment, then the ability for the physical force to bend the virus around something that is a bit too long often can prevent the bending from happening. But that's about as much as I can tell you. It's not my area of expertise, but it's basically not fully understood anyways, but it's understood to a larger extent than I can say.
No, well, thank you already. Okay. But so from what you're saying, it just like flows along like regular pathways.
Yeah, so a lot of the trafficking is pre-existing trafficking that the cell uses for other things anyway. So the virus is not only using the cell to make copies, but it's also using its normal ways that it traffics its own protein that it needs. It's using those traffic lanes that already exist to get itself sorted out.
All right, and next up I see Elka.
Yes, thank you very much. That was really interesting. We're talking very, very fast. I was writing furiously. Yeah, I know we've compressed your time. I don't know if you can answer this question, but I'm wondering, has there been any case of a known natural propensity for immunity to COVID-19 that is without an injection?
So there are some rumors that perhaps it's the case. For me, to be entirely honest, I've suspected it could be the case early on. So what you need to know now, because there's so many asymptomatic infection, what you need to do is you need to meet, well, so many, because there are such thing as a symptomatic infection, you need to make sure that the person is not, it's really a pre-existing immunity, not an immunity that came from exposure with no symptoms. Right. And to establish that now would be very difficult because you'd have to find a population that's been completely untouched with certainty with COVID. What you would have maybe hoped to be able to do is maybe look at blood samples that come from, pre-COVID and look for any sign of immunity there. And the problem is then the other question is, how do you look for immunity? And it's not as simple as you might think. So what people typically do is they'll take the virus, they'll take the kind of cleaned up blood, let's just put it, the cleaned up plasma, let's put it that way, and then try to see if it can neutralize the virus. Or they might look for specific genetic makeup of the antibodies that they find to see if they have similarities. And so even establishing whether a person has pre-existing immunity is not that simple if the pre-existing immunity is not that high. So it's a very complex question, and I don't think there will ever be a clear answer to it, but there could be more and more information that comes out. At this point, it's not something we can exclude, and certainly early on, I started to believe that perhaps there was pre-existing immunity in Japan in particular, because so my partner lives in Japan. I'm normally go back and forth, but now because of travel, of course, I'm in Toronto. And when we exchange how things were, the amount of measure that they take are much smaller, much more reduced. So for example, they can really bring their numbers down. Like they can start seeing a decrease by just closing restaurants at 8 p.m. Until like they still have today, restaurants still open. And you can see right now their case counts are dropping by 5% a day. So it really does seem that it seems to require less effort to have a much more significant outcome in Japan, for example, than in Toronto. But you could point to other factors. One thing that's possible is that they don't have giant house parties and 100% of people wear masks 100% of the time, which is not something I see when I walk out of my door here in Little Italy and people are having nice, fun, together ice creams or gelato on the sidewalks with their masks off. So it could also be just, they're listening better and collaborating better with the instructions. So it's hard to say.
So, and we haven't had enough people die asymptomatically that we could even do a genetic exposure. Like you can see sometimes you have people, like we know with cancer, the easiest sort of generalized kind of example are people who've smoked all their lives, 900 cigarettes a day and they die and they've never had lung cancer.
Yeah.
Then you have other people who just the slightest amount and they go off. So there has to be a genetic component in there.
Yeah, but there's a difference between a genetic predisposition and pre-existing immunity. So what causes you to not have symptoms could be because you have prior exposure, but it could also be that the sum of all your immune encounters so far in your life, never with SARS-CoV-2, but with all sorts of other viruses, you end up with a better immune status, right? If you compared somebody who's immunocompromised to someone who's not, these two people will not have the same outcome when they get infected with SARS-CoV-2. So this is not a case where it's your genetic, but rather just the sum total of all the infections you've been exposed to over your life. And that's a bit different, right? There's two components here. There's like your genetic predisposition and then your built up immune experience over time. And those two could be different. And there's also genetic basis for immunity dysfunction.
Yes. Yeah.
So there's that triple, so that's that other way that it could be. So there's so many ways you could explain a much less severe outcome.
All right. I think we probably have time for one more, and Carol put one in the chat, so I'll read it out here. Carol asks, is it the case that these types of viruses have historically become less deadly over time? Because, for instance, it's against the virus's evolutionary benefit to kill its host. Is there any hope that this could happen with COVID?
Well, that's a very interesting question. I hope not. And here's what I mean by that. For it to adapt that well, it would have to transmit heavily so that it can adapt. I would rather it not do that. I would rather it just go away, frankly. So the thing is, yes, it is generally true that viruses try to tend towards low lethality, so low symptom, high transmission. And in fact, typically, it's happened so rarely that you have high transmission and low symptoms that basically, it's so rare that you can have a virus that's very deadly and highly transmissible that I think we kind of all hoped that kind of couldn't happen somehow. It was maybe wishful thinking. I think SARS-CoV-2 has a very interesting combination where it's basically very transmissible and it can be quite deadly. I hope it will not adapt. I hope we will stop transmission to a sufficient extent that it never has a chance to, and that the vaccine will be able to wipe that sucker out. I don't know how lucky we'll be with that, because it does require us to not think about just ourselves, but to make sure to eradicate it planet-wide. And so that's where we can hope that people will think more globally.
Okay, thank you very much. Can I ask one short follow-up question? I don't know how long the answer will be. I remember that's okay. Thank you for that answer. I remember in virology class at university, they were talking about how SARS had mutated to become less virulent and had basically become, and I believe the swine flu as well, had become almost a seasonal flu. So I think that there were some discussions and hope in the beginning, like as per my question, that this might happen with this virus as well. So you're saying that that would not be a good thing for this virus to do, to follow that pattern?
So it would be great if it just turned into another common cold.
Right.
I'm saying that for that to happen, it needs to acquire a lot of mutations. And that means that our vaccines will not have worked, and it has a chance to still transmit heavily. And I hope that's not going to. So I'm hoping it never gets given the chance for that to happen. So of course you would imagine that it would go in that direction, but don't forget, there is that tendency for the virus to want to go in that direction. But if, for example, that direction that's open to it as a trajectory is blocked, for example, by the vaccines, right? So maybe the vaccines target viruses that have a certain set of properties. And what if that set of properties is the pathway to becoming less lethal or less infectious? It's possible that the virus now to get there would have to go all the way around, and that maybe makes it more difficult. So it's important to understand that by vaccinating people, we're also really changing the options that are available to the virus in terms of the directions in which it can escape. And so it's important to consider what impact we have on the ecology of viruses as they evolve.
So the vaccine would interfere with its natural evolution?
They always do.
Yeah. Okay, great.
Yeah.
Thank you. Thank you very much for the explanation.
Vaccine. So yeah, yes. It would be interesting to see what happened if we stop vaccinating. Would the flu coalesce into a super strain that is very undeadly and very highly transmissible? Who knows, right? We'll never really get a chance to find out, because a lot of people will have to die before we get to that, right? And so that's the problem. To let the virus evolve needs to let it spread freely, to really get a chance to fully explore the landscape, and that's usually causes a lot of death. It could also make it a lot worse before it makes it better. So sometimes that potential barrier to get to the other side of the rainbow is not a pleasant in between. Okay,
great. Thank you very much.
All right. Thank you again, Catherine. If people want to unmute and give a little round of applause, or even if they don't, you can just see them. So, yes. Good.
Thank you, Catherine.
All right.
Thank you.
Coming a bit of a party.
It was great.
Yes, we really appreciate it. Such an interesting discussion on what is, of course, become a very hot topic recently. So all the more information to arm ourselves with as we go and discuss it with other people as well.
Yeah.
Yeah.
So thanks for having me again. And if you have questions, like I don't know how you run these, but if you want to run them another 10 minutes, I'm happy to stick around and answer more questions. You're good? Okay.
I guess I don't see any any other. I mean, certainly I am also happy to stick around if anyone else has questions, but I'm not seeing any in the chat.
Okay. Can I ask something? A quick one.
Sure.
I'm almost 80, so I get the super flu. You know, the flu. Yeah, you know, the extra one. But with these injections, they all seem to be the exact, one size fits all. So it makes me very uneasy. Well, I'm very uneasy about this whole process, but that's a whole other issue.
Well, so here's what I can say about that. The problem with not having mathematical modeling to inform our decision. So when we launch a spaceship, right, we do it because we know the equations of motion and we know exactly what force to apply to get it exactly where we want. The thing with immunology or virology is that there are no equations, which means the only way we can know what the outcome will be is to actually carry out the experiment, right? Okay. And so to be able to answer your question, I would have to vaccinate and humans are very varied, a lot of underlying condition. I would have to vaccinate a whole pile of people in your age group, and then I could give you the answer. What I can say is that when they did the evaluation for the vaccines, they wanted an approval. They wanted to go for like the middle of the pack. And so they want for kind of middle of the age group people. And so there isn't as much data. And so right now we're vaccinating based on that one protocol that was established in that one type of population. And anybody who tells you they have an answer are lying, because it's not a linear system, right? We don't know. We can't say if you double the dose, you get twice the protection. You can, if you remember when they did the one of the vaccine, I can't remember which one, when they were doing the test, the second dose, they accidentally underdosed. And they found out actually that the underdosing actually created a higher protection than the higher dose for the second. And so it's very counterintuitive, but the immune system is a complex system that has cells that influence other cells that influence other cells. So to be able to understand how these networks, these complex systems work, we need a mathematical model to be able to predict it. Otherwise, unless you try exactly that protocol in a large number of people, all you have is nonsense and random numbers. So nobody can tell you that answer unless this is done in a large enough scale. And that's why it makes me angry about biology. And that's why I do what I do. I'm trying to develop equations so that we can predict and not just hope for the best or try it in people.
And also, we only know what they actually tested for. We don't know what else it does or doesn't do, because they didn't test for other stuff.
Right. So what I would say about the SARS-CoV-2 vaccine, I know a lot of people are very concerned about that vaccine and how rapidly it was developed. A lot of people have gotten this vaccine by now, like a lot, like I think it's 40 million got the Pfizer vaccine so far or something like that. Let me check my numbers. I have them right here. I don't want to lie to you. I prepared those specifically for that question. So basically, a lot of people have received the vaccine so far. So if the vaccine itself was dangerous, we would have had serious problems already, right?
Not just as well.
I'm sorry?
I was going to say, you may not have the symptom yet, but it doesn't mean
no, it would have. So it happens immediately, right? So first question you have to ask is the vaccine alone safe? So when I get the vaccine, are things okay for me, right? That is a done test. We've had, so by this time you see 35 million. This is just for 8 countries. 35 million with Pfizer, 26 million with Moderna. So we've got a very large swath of the population that's received these vaccines. They are safe. The next question,
know that's not my problem. Like what I think of as thalidomide. You know, thalidomide was only tested for one thing, and that is, was it going to cure morning sickness in pregnant women? And it got results like this. It was brilliant. It was like 100%, like almost right away. And they didn't test it even for one full pregnancy cycle. So but this is what terrifies me.
But here we have done it. So we're talking about the vaccine alone. So what you're talking about, the vaccine in combination with the condition, right? And so here, if we're talking about the vaccine being combined with the virus exposure, that's where there could be complication. But we're having a mounting number of people who have been vaccinated and also then were exposed to the virus, and we've yet to see complication. And by the time, for me at least, by the time I'm going to be offered a vaccine, that number is going to have been sufficiently large that I'll have that last concern. So when they launched the vaccine, the SARS-CoV-2 vaccine, you could feel safe about 1 and 2, but you could not feel safe yet about number 3. But with every day that passes, we're building more and more data on number 3. And for me, at least, by the time anybody, sorry.
That's still not answering my question. I'm not being clear. The vaccine is being tested for A, B, C, and D. All right. It may take 2 years for G to occur, and G could be very serious.
So it depends what's in your vaccine, right? So in this case, we know that this vaccine contains this mRNA. We know what mRNA can do. So the mechanism, so all vaccines are not created equal.
Right.
And I'm not telling you to be always trusting of vaccines. I'm saying for each vaccine, you need to look at what is it made of and are there reasons to be worried? I have looked at what's inside that vaccine. I think it is the right decision to go forward with vaccination, and I don't expect there will be serious problems. And I can't see a mechanism, because you also have to propose a mechanism. What of all the things that are inside, if you have to be super creative, and that's a question you want to ask a virologist or an immunologist. What you want to ask them, don't ask them about pandemic control. They don't know about these epidemic numbers, right? But you should ask them about this stuff. If you had to be creative and you were forced to come up with a way that this vaccine could go terribly wrong, could you think of any mechanism that could cause that, right? So you have to look, these are our mRNA vaccines. Could you see a way? And I really honestly can't think of one.
Okay, that's good. Yeah, thanks.
All right. Catherine, if you're willing to take some more, I saw that George had a question.
Hi, Catherine. I want to go back to the seasonal flu vaccine. Every year I take the seasonal flu vaccine, and after I turned 65, I've got the high dose vaccine. Now, what happened is that this year, when they started doing it in October and November or whatever, the high dose and the regular flu vaccine were not available everywhere, and it was very hard to get it. And then eventually I got the regular vaccine, not the high dose vaccine. But the question is, around that time I was chatting with a friend of mine, and who knows more about biology and medical issues than me, and explained that the difference between the high dose and the regular one is not simply a matter of the effectiveness, the amount, and so on, but the composition, and that the regular vaccine consists of 3 strains. I'm going to use the word loosely, while the high dose is 2 strains of in a higher amount. And maybe I don't understand what's the thinking behind it. Maybe you can explain that. Maybe somebody else is curious about that too.
I could similarly not tell you, because number one, I've never looked into the 2-dose vaccine, so I'm actually not even aware of what they put in it. But the other thing I would say specifically, especially if you're concerned this year, is that if you're staying indoors to prevent COVID, you have no reason to worry about the flu. And in fact, when I saw everybody make a rush for the flu vaccine, my partner and I were basically laughing, because we thought, like, if you think there's any chance you're going to get the flu, you should be a lot more worried about dying of COVID, right? So by the time you've got the flu, you've really hung up with a lot of people, because it's much less transmissible than COVID. So I wouldn't worry too much about that. If you're staying indoors and continuing to distance with people, just forget about it. And the next season will come around, and then you can get next season's flu vaccine. Because by the time you're vaccinated for COVID, it'll be our bright summer, and then you can worry about the next year's strain.
Just to be clear, I wasn't worried. I was just curious.
So in terms of your question, though, what I can say is that the only way we know why it's effective is because we've tried it. So I'm sure they tried different protocols and found out that this worked best. And I'm sure they will give you an explanation for why they think that's true. And that explanation might even be true. Okay. But as I said, in the absence of a mathematical model, all we can do is try stuff, find what works, and tell everybody to do it. And you can see that's largely how the COVID thing has been approached. But you'll be surprised how much of health works exactly this way. Try the stuff out, it works, tell other people, and that's what we're doing from now on. And we don't always know why it works.
Okay.
But we can make stuff up.
Okay.
We can suggest reasons.
Okay.
Carol, you had put another question in chat. Do you want to say it yourself, or shall I?
Oh, sure. Yes. Oh, I can do it. Are any of the pharmaceutical companies working on clinical trials to determine the proper dose of the vaccine for children? Because I'm one of those parents that does not want to send my child back to school in person until he's been vaccinated. So I know that, of course, they were focused on seniors, as appropriately they should have been, and that the children will very likely come last. But I'm hoping that they will have something for them before September. Am I dreaming, or is there any possibility of that?
No, I think trials are maybe, and I'm not certain here. I'm sure a very quick Google will tell you, because the drug companies will put out press release. So it's not fake news. You can find from the drug companies themselves, they will have said that they're starting trial in children or something. I'm pretty sure at least one company would have started those already. And so whether these will be available by September, your good is as guess as mine. The only difference, so what happens is that they have to get enough, they have to enroll enough people in a trial for that to be a valid data for approval. And so I don't know how much data they will have by September. And I don't know even, to be honest, whether the vaccine has been approved in Canada so it's not approved, it's under emergency use authorization. And I don't know if, which population will not be allowed under that emergency authorization. So I don't even know whether children could. I mean, for one thing, they're not available right now at all for children. Who knows, probably by September, I don't know if even I will have been vaccinated. So I don't know that you can really hope that your children will have been. But, fingers crossed, all of us.
Okay, thank you.
Okay, may I ask one question? It's something that you had brought up at the beginning, but so how do you make the jump from physics into biology?
Frustration, I guess. No, so no, it wasn't, because I didn't know how frustrating it would be when I entered the field. I don't know that I would have if I knew now what I know. So basically what happened is I was interested in intelligence, artificial intelligence, primarily, actually. So I wanted to go into computer science, and the computer scientists said, no, the physicists are much more useful. And I was like, okay. And I happened to really love physics, so it worked out. So I trained in physics with the intent of going back to artificial intelligence. And then I started working on intelligence itself, like neurons and neural network and how it learns. And then I found out that this is such a complicated system that at the scale at which memory or intelligence emerges, you have so many neurons that people kind of cease to understand the system. But there's a system at which it happens in a much simpler way, which is the immune system. It's able to adapt, to learn, and to memorize disease that it's encountered before. So that was my entry into immunology, which rapidly got me pivoting. And then that's when SARS hit. So strangely enough, so this was in 2003, that's when I started doing virus research. So all of this happened while there was this pandemic in Toronto of the first SARS, and that actually corrected my trajectory in life in a sense. So that the first SARS is credited for my involvement now in the second SARS. But basically, that's what actually triggered it, is that I was interested in studying intelligence, and only the immune system had a scale at which you could actually study these mechanisms. And then you immediately want to know how it's being applied to a virus, and then that's turtles all the way down.
Awesome.
Yeah, well, I guess we're certainly glad that you found that path today, for sure.
Thank you.
All right, I'm not seeing any more questions. Yes, deletingly.
Thank
All
you
right.
so much, Catherine.
Yes, thank you. Thank you again.
Catherine, there's a question in the chat. Somebody's, or not a question, a comment that the regular flu strain, the regular flu had 4 strains, and
it's not quite true,
other
right? So you mean the vaccine, and
yeah.
It's not quite true. There is the quadrivalent and the trivalent. So it's possible that your doctor gave you the trivalent, and then you'd have the 3 strains, but the quadrivalent is also available in Canada. So it depends on which of the vaccine, and it's not just the high dose and low. Oh, maybe you're right.
It's not my comment. Somebody wrote it. I don't know. I'm asking.
So a mystery is a mystery. Basically, I think that is possible that the high dose I would have thought it was the other way around though, but it's possible that all the high dose happen to be the trivalent and all the low dose, the regular dose would be the quadrivalent.
Why?
That I'm not so sure how the products get, because that's come down to how the products get distributed. I know what vaccines we have available in Canada. I don't know who gets which.
Catherine, that was the basis for my question. That's why I asked the question, which it puzzled me that the high dose had fewer strains than the regular dose. That's why I asked.
I think according to the comment, they have it the other way around.
No, the regular has.
No, that's true. The regular has 4, and the high dose would have 3.
Yes, that was the reason I asked the question.
But all you'd be missing then is the second flu B. So the second lineage of flu B. So if it's not a bad flu B year, and this year is not even a bad flu year, but if it's a bad flu B year, that's where it could be a problem.
All right.
Does that always happen? Are there always some with 3 and some with 4?
That's not my expertise. I couldn't tell you for sure. I didn't even know about this, the high dose, low dose not being the separation.
Because I want to know what I have to ask, before I go, because.
You can ask them. Yeah, of course, your doctor will know and will be able to tell you. But if they formulate the high dose as 3 and the low dose as 4, you won't really have a choice anyways.
Yeah.
Yeah,
It's
I'm
not
a
like types of shampoos. You don't get as many choices.
I'm a pharmacist, and
Oh, there you go.
I put the comment. So the regular flu shot has 4 strains. It's the quadrivalent, and the high dose flu shot has 3 strains. But I believe it's just higher doses because as for seniors, people age 65 and older, their immune systems tend to be weaker, and they need a higher dose of the component. So that's why they need a higher dose to get that immune response.
So why don't they get 4 strains higher dose as opposed to 3 strains?
Yeah.
What's the trade-off? Is really
that I don't know. Sometimes we accidentally give the regular flu shot to our seniors. Sometimes that mistake happens when we're in hospital, and we don't really think of it as a big deal. You're getting more coverage, but you're just not getting the higher dose. I don't know. You'd have to ask maybe the people who develop the vaccines why they do it. Maybe it's
and most likely it's just so it's different manufacturers oftentimes, right? So the manufacturer, they might just have different protocols. So it used to be that all the vaccines were trivalent. The quadrivalent is somewhat new. And so it's possible that that manufacturer is keeping its old standards, because to introduce one more strain would mean going back to trials to see whether adding one at high efficacy had the same efficacy on the other 3 strains. And so you could see it's, again, that whole problem that unless we do the test, we have no idea. So it could be that there's 3 simply because that's all they tested at the time, because at the time, all the competitors were also trivalent. It could be as simple as that. It might not even be an immunological basis for it.
All right, this is a group of scientists, science lovers, and skeptics. So this is an intervention. I'm cutting you all off.
All right.
All right. Once
so thanks
again,
for having me and
thank
for
you.
the very
Thank
fun
you.
conversation.
Thank you.
Thank you. Take care.
Stay
Thank you.
safe, everyone.